The virus envelope protein GP120 that binds to the receptors on the CD4 cell and fuses with it with the GP41 protein and penetrates into the cell and coils, and viral DNA, enzymes and protein are released into the host cell cytoplasm. So this is the HIV virus, and this is the GP120 protein which binds to the CD4 cell, and then the second protein GP41 on the HIV virus fuses with the CD4 cell, and then there occurs penetration into the cell and coiling of the virus. See this, I showed it in purple, penetration and coiling, and then the viral RNA, enzymes and protein are released into the cytoplasm of the CD4 cell. So GP120 binds to the CD4 cell, and then this fuses with the help of GP41 protein with the CD4 cell, and then penetrates and coils into the CD4 cell, and viral RNA, enzyme and protein are released into the host cell. The CD4 cells have co-receptor for the entry of this virus into the cell. The two major co-receptors are 5-CCR and CXCR4 receptors. So this is the CD4 cell. It has two receptors, 5-CCR and CXCR4 receptor here, that combines with the virus. HIV envelope protein interacts with the CD4 and chemokines and receptor cytokines cause increased cellular activation and HIV replication. Virus is cytotoxic to the CD4 cell. The virus binds to the CD4 cell receptor here with the help of GP120 protein, and then fuses with it with the help of GP141 protein, and then coils into, penetrates into the CD4 cell, where the viral genome, proteins and enzymes are released into cytoplasm of the CD4 cell. So what happens in the cytoplasm of the CD4 cell? The viral reverse transcriptase then act on the genomic RNA and convert into double-stranded DNA, which then, along with the integrase, enters into the nucleus of the CD4 cell through the nucleopores, and this DNA is integrated into the DNA of the CD4 cell, and then mRNA comes out for the protein synthesis, and viral genome comes out and leaves the cell after budding. So I repeat here, in the host cell cytoplasm, viral DNA, reverse transcriptase acts on viral DNA, converts into double-stranded DNA. This is reverse transcription. The reverse transcriptase first forms single-stranded DNA, and then it forms double-stranded DNA, as I shown here, and then it enters through the nucleopores along with integrase into the nucleus and integrates with the CD4 cell DNA with the help of the enzyme integrase, which also enters the nucleus. This provirus DNA remains transcriptionally inactive or latent for some time until it activates to replicate the HIV cycle. So how it releases the mature virion? The other enzyme, protease, cleaves the gag-pol from the provirus and converts it into mature virion. See here, this one, protease cleaves the gag-pol to yield the mature, from provirus to yield mature virion. So what does gag do? Gag encodes the protein that forms the core of the virus. So I repeat, viral particle is formed by the assembly of HIV protein, enzyme and genomic RNA at the plasma membrane of the cell, and the enzyme protease cleaves the gag-pol precursor to yield mature virion.